Alright, hello everyone. My name is Tessa Firth, and I will be presenting my biochemistry project on the metabolic process known as glycolysis. The structure that just popped up to the right here is a biomolecule known as glucose. We'll talk more about that within the presentation, but just to start off, it is the main biomolecule that starts glycolysis. Alright, so to introduce glycolysis and to start off, some of you might be thinking, what is glycolysis? So glycolysis is one of the metabolic processes involved in cellular respiration. And cellular respiration is how energy is made within your body. So in practical terms, let's say you're going for a run. You're getting the energy that it takes to run and to complete the process of running and the actions that it takes through cellular respiration. It happens several times when you run. There are several processes in cellular respiration, but we'll just summarize it in three main processes. So that is glycolysis, which what we will talk about today, the citric acid cycle, also known as Krebs cycle, and the third being oxidative phosphorylation, which was where most of the energy and ATP is made. So the diagram below is a very simplified version of cellular respiration. We have the cytosol out here, which is where glycolysis happens, and we have the mitochondria, which is where the citric acid cycle and oxidative phosphorylation happens. So where does glycolysis happen? Glycolysis occurs within the cytosol of a eukaryotic cell. Some of you may have heard from the cytoplasm and not the cytosol, because the cytoplasm is studied, I feel like, a lot more than the cytosol, but they are, however, very different, as the cytosol can be referred to as the cytoplasmic matrix surrounding the essential organelles of the eukaryotic cell. The cytoplasm, however, is the entire fluid component in the eukaryotic cell. As glycolysis takes place in the cytosol, the products travel into the mitochondrial matrix for further processes known as citric acid cycle and the inner mitochondrial membrane for oxidative phosphorylation. As you can see in the diagram to the left here, the beige is the cytosol, which is where glycolysis is happening, and then the mitochondria is the yellow and the dark red. So we have the inner mitochondrial membrane here, and then the mitochondrial matrix on here. And then the diagram to the right is simply illustrating the differences between the cytosol and the cytoplasm, which is just what was talked about. So what biomolecules are involved in glycolysis? Well, there are several biomolecules in this process, around 10 actually, but the main biomolecules which are essential to the process are just going to be the four that we talk about on this slide, which is glucose, pyruvate, adenosine triphosphate, and one of the electron carriers known as NADH. So to start talking about glucose, it is the beginning biomolecule of glycolysis, composed of several monosaccharides originated from sugars, glycerol from fatty acids, and amino acids which are consumed by organisms on a regular basis. The chemical formula for glucose is C6H12O6. And then we're going to move on to pyruvate, which is an essential biomolecule in biochemistry, known as the product of glycolysis, as one molecule of glucose is broken down into two molecules of pyruvate for further usage of energy through cellular respiration. So pyruvate, eventually after glycolysis, gets converted into a biomolecule known as acetyl coenzyme A. You might have heard it referred to as acetyl-CoA, which is the starting enzyme in Krebs cycle. The chemical formula for pyruvate is C3H4O3, so that's almost half of glucose. So then we have adenosine triphosphate, ATP. This is a 3-phosphate molecule containing one adenine group and sugar, another product of glycolysis, and a molecule that helps the process happen. We'll talk more about ATP in the further slides. So then we have the electron carrier, NADH, which stands for nictoamide adenine dinucleotide plus a hydrogen. It is essential as it is an oxidative cofactor in the production of energy. So the diagram below really summarizes, again, cellular respiration. However, we're just going to be referring to this part up here, which is glucose and pyruvate. So then when we were talking about glucose, we talked about glycerol, amino acids, and sugars, which are the origins and originate glucose. And we have pyruvate down here, which is what is the product of glycolysis, one of the products. Okay, so we're just going to talk a little bit more in depth about glucose here. So what essentially is glucose? In practical terms, glucose is the main sugar source provided in the body, coming from foods such as proteins, lipids, carbohydrates, and they're being consumed by the organism. So glucose travels through the bloodstream and into living cells to be further converted into pyruvate for energy in the process of metabolism. In biochemical terms, glucose is a simple sugar, so that's an aldohexose monosaccharide, and one of the main carbohydrates within your body. There are two sister molecules to glucose known as galactose and fructose, which all have the same chemical formula as glucose, C6H12O6. So both galactose and glucose have the exact same structure. They are aldohexose sugars, where fructose is a ketose. So galactose and glucose are structure defined by only one component of their structure, where OH group on carbon 1 on glucose is pointing down, and it's pointing up on your galactose, you can see here. So that's the only difference between the two. Glucose is the most abundant sugar molecule than fructose and galactose, as organisms utilize glucose more frequently than fructose and galactose for glycolysis, because cellular respiration essentially happens many times in your body per day. So up here we have a more advanced structure of glucose, C6H12O6. You see your oxygen molecules in red, your hydrogen in yellow, and then your carbon molecules in gray. Alright, so what exactly is energy? So with all this talk about energy that we've been saying, ATP this, ATP that, what exactly is it? So energy in biochemistry can be referred to as adenosine triphosphate. Adenosine meaning that there is an adenine group. Let's pull that up here. So your adenine group is right here. And then we have a ribose sugar group attached right here. And then triphosphate of the adenosine triphosphate means three phosphate groups. That's right here. They're all attached here. So ATP is very essential to all living organisms and is needed for processes such as nerve impulses, DNA replication, protein synthesis, muscle contractions, and more daily movements and activities that happen in your body all the time. So in practical terms, the way to consume ATP is through food and oxygen. You need to have both. You can't have all this food in the world and then no oxygen, and you can't have all the oxygen in the world and no food. You need both. So you need to have both to produce ATP. So in glycolysis, there are a net gain of 2 ATP, which is what we're going to talk about a little later in the reaction, and is the first pathway in cellular respiration where ATP is used and made. Okay, so what exactly is the process of glycolysis? We've talked a lot about what glycolysis is, what glucose is, what ATP is. So let's just summarize in very simple terms the process of glycolysis. So you need glucose to happen for this. You need glucose for this process to happen, sorry. So you have a 6-carbon molecule starting this reaction, being converted into a more reactive form of itself known as glucose 6-phosphate. So this little conversion here requires ATP. Glucose 6-phosphate then gets converted into fructose 6-phosphate, where fructose 6-phosphate later gets converted into fructose 1,6-biphosphate. So it gains a ATP. Again, you're investing in an ATP molecule over here. So it gains a phosphate group. So this 6-carbon sugar gets split into 2 3-carbon molecules, one of them being dihydroxyacetone phosphate, also referred to as DHAP, and glyceraldehyde 3-phosphate, which is also referred to as G3P. So eventually all of our DHAP is going to be converted fully into G3P. G3P is then converted into 1,3-biphosphoglycerate. So that yields one of our electron carriers known as NADH, which we'll talk about in the more detailed steps of glycolysis. So 1,3-biphosphoglycerate is converted into 3-phosphoglycerate. So you're going to lose one of its phosphate groups. This yields an ATP molecule. So our phosphate group from 3-phosphoglycerate is going to move to our second carbon, so that's going to be 2-phosphoglycerate. And then the 2-phosphoglycerate gets converted into phosphophenols. So then phosphophenols gets converted into pyruvate, so it's losing a phosphate group, which is on our second carbon right here, and that again yields an ATP molecule. So you can note here that this reaction below the yellow line is happening twice. So after the splitting of the 6-carbon into 2 3-carbon molecules, we're going to have a reaction happening twice. So that's going to yield a net of 4 ATP, but 2 of those ATP are going to be used as an investment in our reaction of glycolysis, hence why there's only 2 ATP as a product of glycolysis. We'll talk about that a little later in the more detailed steps. Okay, so I know I talked about that process of glycolysis really fast, so we're just going to take it in 10 simple steps. So the first step includes a phosphate group being converted from ATP to ADP. So you're going to add a phospho group catalyzed by the enzyme called hexokinase. So your hexokinase is going to catalyze the reaction from glucose to glucose 6-phosphate. So you see we have the ATP molecule plus glucose, and then we have our phosphate group being added onto our glucose molecule. So it's glucose 6-phosphate plus an ADP molecule. You see the structure of glucose molecule. I have the phosphate molecule down here. So this yields glucose 6-phosphate, which is the more unstable form of glucose. Alrighty, so our second step of glycolysis consists of our glucose 6-phosphate molecule being converted into fructose 6-phosphate. So that's what's happening in this reaction down here. So this is catalyzed by an enzyme called phosphoglucose isomerase. So essentially, the isomerase part is kind of illustrating that fructose 6-phosphate is a product of the isomerization of glucose 6-phosphate. So fructose 6-phosphate is the isomer of glucose 6-phosphate. As you can see down here, these structures are quite similar. Alright, so the third step of glycolysis is catalyzed by a very essential enzyme known as phosphofructokinase. We can call it PFK for now, just because it's a little shorter. So this reaction, catalyzed by PFK, involves one of the 3 phosphate groups from adenosine triphosphate molecule to be transferred onto fructose 6-phosphate, producing fructose 1,6-biphosphate. So that's when our phosphate group is going to be added to our first carbon molecule and our second carbon molecule. So I'm just going to pull up a little illustration here. So this really illustrates what activates this enzyme to catalyze the reaction. So low levels of ATP lead to a fast production of ATP needed for cellular respiration to occur, hence glycolysis. So if the cell is low on ATP, it will get more ATP from an ADP molecule through the reaction of ADP plus ADP yields ATP plus adenosine monophosphate, which is right here. So high levels of adenosine monophosphate in the cell activate PFK to the cell being starved for energy. So when we have low levels of ATP and high levels of AMP, phosphofructokinase catalyzes this reaction. But if the cell has a high level of ATP, and it inhibits right here the production of phosphofructokinase, which makes sense since if there's already enough ATP within the cell, glycolysis really doesn't need to produce AMP. Okay, so our fourth step of glycolysis splits our 6-carbon sugar molecule, fructose 1,6-biphosphate, into 2 3-carbon sugar molecules. So the first one is dihydroxyacetone phosphate, DHAP, which is right here, and our glyceraldehyde 3-phosphate. So these 2 sugar molecules are isomers of each other. As you can see, they're very similar in structure. The only difference is the double bond between these 2 carbon molecules right here. Alright, and our fifth step of glycolysis, we're halfway there, involves all of the DHAP, which is the isomer of our glyceraldehyde 3-phosphate, to be converted fully into a glyceraldehyde 3-phosphate molecule for the sixth step. So, the sixth step of glycolysis involves an oxidization of the 3-carbon sugar known as glyceraldehyde 3-phosphate. So this is oxidized by our cofactor, remember that we mentioned in the earlier steps of which biomolecules were involved in glycolysis, NAD. So there are high energy electrons removed and picked up from this oxidization by the electron carrier molecule NAD+, thus producing NADH and H+. So these are 2 of the products of glycolysis, and they're yielded in this reaction, in this step, so just remember that. So this reaction releases ATP that is used by phosphorylating the oxidized form of glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. So just ignore steps 7, 8, 9, 10 right now. Focus on this right here, because that's just when our glyceraldehyde 3-phosphate is being oxidized into 1,3-biphosphoglycerate. So going from this to this, yielding our NADH plus H+. Okay, so step 7 of glycolysis involves 1,3-biphosphoglycerate giving one of its 2 phosphate groups to an ADP molecule. So we see here what I keep saying ATP to ADP. So you kind of see in detail here what is happening when we go from ADP to ATP and ATP to ADP. So you're gaining and you're losing a phosphate group. So you go from a diphosphate group with ADP to a triphosphate group ATP. So this ADP molecule is converted into ATP. The ATP is then used to convert 1,3-biphosphoglycerate into a 3-phosphoglycerate molecule. So that's happening right here. and it is catalyzed by an enzyme called phosphoglycerate kinase. So you're yielding one of our ATP molecules. This is where one of our ATP come from when we're having our net gain in the products of glycolysis. So one of our ATP molecules comes from the 7th step of glycolysis. All right, our 8th step is very simple. So the 3-phosphoglycerate molecule is from the 7th step is converted into a 2-phosphoglycerate molecule, which is its isomer. So this is happening right here between step 7 and step 8. So we have our 3-phosphoglycerate being catalyzed by an enzyme called phosphoglycerate mutase, and that's moving our phosphate group from our carbon, our 3rd carbon to our 2nd carbon. So you see the phosphate group in the red, replacing the hydroxide group over here, so they switch around. So that makes 2-phosphoglycerate. And then we have that molecule right here, 2-phosphoglycerate again, so just in another form, so you can see its actual structure. So they are isomers of each other. Okay, so the 9th step involves 2-phosphoglycerate molecule losing one of its water molecules. So I'll just show you that little reaction down here. So as the 2-phosphoglycerate molecule loses its water molecule via the reaction catalyzed by the enzyme enzolase, it forms a very unstable molecule known as phosphophenylpyruvate. We're going to call it PEP right now just because phosphophenylpyruvate is quite long. So in the last step of glycolysis, this unstable molecule will lose its only phosphate group. So our phosphate group's right here in pink. Okay, so our 10th and final step of glycolysis involves the unstable molecule PEP phosphophenylpyruvate losing its only phosphate group. So that's in our little diagram down here. So this phosphate group is then given or donated to ADP, which makes another molecule of ATP. So this is where our 2nd molecule of ATP is gained. So remember, we have 2 molecules of ATP made at the end of glycolysis. This is where the 2nd one is made. So as the PEP molecule loses its phosphate group, it becomes pyruvate, which is the end product of glycolysis, a very, 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 very important molecule in glycolysis. This is what is our product. So pyruvate floats to the mitochondrial matrix to be later converted into an enzyme, acetyl coenzyme A, for the starting enzyme of Krebs cycle, citric acid cycle, to make more ATP. So this is our last and final product of glycolysis. Okay, so what are the end products of glycolysis? What is the end product of glycolysis? So there is a lot of products in each step and yielding of molecules, so I'm just going to summarize it. So the end products of glycolysis are 2 molecules of ATP, 2 molecules of pyruvate, since from step 6 to step 10 happens twice, we have 2 molecules of pyruvate, 1 molecule of water, 2 molecules of NADH, and an hydrogen ion. So these only occur in aerobic respiration in the presence of oxygen. So aerobic means oxygen present, anaerobic means oxygen absent. So the product of glycolysis in the absence of oxygen is lactate. So that's why I have a marathon runner up here in the corner and a weightlifter down here. So our marathon runner is going to be using aerobic respiration because that's where most of our ATP is made. So aerobic respiration is glycolysis, Krebs cycle, oxidative phosphorylation. So you need all of your ATP via oxidative phosphorylation, Krebs cycle, and glycolysis. That's where most of the ATP is needed. So when you're running long distances, like 42 kilometers, you're going to need a lot of energy. So that's aerobic respiration. Anaerobic respiration, or anaerobic glycolysis, occurs when you need ATP immediately. So this guy he needs energy immediately to lift that weight he's lifting. So this is a maximal effort exercise. So he produces lactate. He produces lactic acid from anaerobic glycolysis. Okay, so what is the purpose of glycolysis? There's a lot going on in this process. What is the purpose of it? Why does it happen? So the purpose of glycolysis is to convert 1 molecule of glucose, so 1 sugar molecule, into 2-pyruvate for later energy usage. So the 2-pyruvate move into the mitochondrial matrix where they're converted into acetyl-CoA, as mentioned before, for the process of the citric acid cycle to make more energy. So there's 2 molecules of ATP made in glycolysis, 2 molecules of ATP made in Krebs cycle, and 28 molecules of ATP are made in oxidative phosphorylation. So oxidative phosphorylation only happens in the presence of oxygen, only happens through aerobic respiration. So glycolysis is very important because it is the beginning step of cellular respiration and the beginning step of all this ATP being made. Okay, so I'm just going to go over some questions that I had when I was learning glycolysis. So the first one is, where do the NADH and the hydrogen ion go after step 6? So they just hang out until glycolysis is over. So no, if in oxygen-present conditions, so in aerobic conditions, the NADH and the hydrogen ion go into the mitochondria to later be converted back into NAD plus for further ATP production in oxidative phosphorylation. So NAD plus is an electron carrier, so it carries electrons in oxidative phosphorylation to make ATP. So the NADH and the H plus are needed for oxidative phosphorylation in later steps. The other question is why are 4 ATP made in the process, but only 2 net ATP are gained as the products in glycolysis? So one of the ATP, remember, are used to convert glucose into its glucose 6-phosphate unstable form, and another ATP is used to convert fructose 6-phosphate into fructose 1,6-biphosphate in the beginning steps. So that's where the 2 ATP are used. The last step, when glycolysis occurs under anaerobic conditions, do products still move on to Krebs cycle? So a simple answer is no, they do not. So glycolysis is repeated under anaerobic conditions, and the NAD molecule stays in a reduced form until pyruvate forms as a product of glycolysis. So pyruvate is used by an enzyme called lactate dehydrogenase, LDH, resulting in a molecule called lactate to be formed, which in turn forms lactic acid. So when you're doing a workout, you're working out, and your muscles start to burn, that's your lactic acid building up in your blood, in your muscles. So you need to rest, you need to breathe in oxygen for that lactic acid to be flushed out. So that you need to go through oxidative phosphorylation to later make more ATP and do more reps of your exercise. All right, well, thank you very much for watching. I hope you learned lots about glycolysis and you can apply it to practical terms as well. Here are a list of my references as well. Thank you very much. Bye.